Well, so hi. Today I'll be talking about muscle, and I think if we're lucky, I'm able to cover pretty much everything about the microscopic nature or the microscopic aspect of muscle in this very lesson. The next lesson, well, it continues with movement, but today we're going to be looking at a very microscopic structure. And what you see over here, that is under the electron microscope, that's muscle, okay? And what we have here is, I arranged this several times next to each other. And what we're going to be doing is I'm going to be labeling this here because what you see here is our so-called sarcomeres. Sarcomeres. How do you actually write this? Sarcomere, that is the basic unit of contraction in a muscle, okay? And as I already mentioned, we are now looking deep into a cell, and this is an electron micrographic image. The question is now, where is a sarcomere here? And what I'm going to do now is I'm just going to label this. A sarcomere goes from here to here, okay? So that is a sarcomere, and that's a contractile unit. And you see that in this case I've got 1, 2, 3 sarcomeres, but actually there are many, many thousands of them going in a row, and together this actually makes a so-called a myofibril. So the question is, where am I going to label this here? So this whole thing here, I don't know, yeah, the whole thing going across, that's called a myofibril. So these are many sarcomeres arranged next to each other. And what you're going to see as well is that there is an alternation of light and dark areas. And this dark gray area here in the middle, this is known as a so-called a dark band. It's a dark band. And of course from here to over here, that is known as a light band. Do notice that there is a pretty dark line here, okay, here and here and here. And these dark lines that you see here, these are actually a part of the light band, and these are known as the so-called the Z lines or Z lines, Z lines, okay? But they look pretty dark, almost black, but in reality they actually do belong to the light band. And now what happens is that when a muscle contracts, there are fibers here, and then the light band, this is the one that becomes smaller when the muscle contracts. So what you see here is now a relaxed muscle, but when it's smaller when muscle contracts. So this muscle here is as a matter of fact is relaxed, yeah. So this will become smaller, and what will happen actually is that those dark bands here, they will move, together, and the light band will become more narrow. Yeah, so that is essentially pretty much all that there is to label. But what I'm going to do now is I'm just going to show this to you over here using a schematic diagram. And what we have here is, again, here is a sarcomere. That's the Z line. So I'm going to label it down here. It's the Z line. And from here to here, that is, of course, one sarcomere. Okay, and here I think you see quite well the difference between the contracted and the relaxed version. So this one, of course, is contracted. The muscle is contracted. And here it is relaxed. The question is now, where is the light band in the first case? Well, in the first case, we're missing something here. Of course, it continues over here as well, right? Yeah, and we need the thick filaments in here, yeah, which I did not indicate. So the light band is from here to here, okay? That's the light band, and also from here, maybe I'm going to draw this a little bit differently, to over here, okay? And you see it is much smaller than from when in the relaxed version, when it's from here to all the way over here. So that's much bigger. So that's the light band. And here the light band, is actually yeah, that's from here to here, that's also the light band. Okay, from here all the way to over here, and you see that the Z line is part of the light band. You also see that the fibers that are involved here actually they do not change in length, and the blue ones here, these are the thick fibers. They're the thick ones, and they have a name. It's called myosin. And the thin fibers here, that is actin. We're going to look at this in more detail later on a little bit. But what you see that is also that over here, where there is the myosin, this is where you have the dark band. So you see that the dark band goes from over here to all the way over here where the myosin is. Okay, so that's the dark band. So the dark band does not change. It does not change when the muscle contracts. The only thing that changes in size is the light band, okay, which is small when it is contracted and which is large when the muscle is relaxed, okay. So and the muscle contracts because those two protein fibers, the actin and the myosin, they slide past each other. So I should actually write for the actin, these are the thin filaments. The thin filaments, okay, so they slide past each other, but they do not change their length as such, okay, that's really important. So muscle contraction happens because the fibers slide past each other. So what we have over here is another picture which shows you a little bit the whole thing from a different perspective. I think I need to change now the color a little bit over here, otherwise you're not able to see what I'm drawing here. You go here from a very large picture all the way to a very small, to a molecular scale. You see over here, the actual muscle. We're going to be talking about this later on. But one of the things that I can already mention, because you have to know it anyway, here there is the bone, and you see that the muscle over here, I call this the muscle, okay, is connected to the bone over a tendon. Okay, that's pretty important. So a tendon is very inflexible, it does not contract, but it's there to join the muscle and the bone together. Okay, so that's the tendon here. So the muscle is actually able to pull on the bone. We would like to talk about this later on a little bit. So what we have then is in here we have an individual muscle fiber. These are actually things that you can see when you eat meat. You are able to see the individual muscle fibers. So that's this one over here. So it's the muscle fiber. Okay, and each muscle fiber has in here many myofibrils. So these ones you already know because I just talked about them. Okay, so these are the myofibrils. Okay, so it's an arrangement of the dark and light bands, and of course you already know what it is from here to here. That is the Z line. Okay. And from Z line to Z line, that is, it says here actually one sarcomere. Yeah? And so you see in here, maybe I'm going to color this in here a little bit. You see this here? That is the dark band, or the myosin. It's another myosin over here. Up, I misclicked. Okay. Yeah, and another myosin over here. So the dark band actually goes from over here all the way to over here. That's the dark band. Do I have to label this? And the light band, that's kind of drawn a little bit badly here, because here it's also like this. Yeah. So this here, that is from here to here, that's the light band. And this here in the middle, that's the dark band. Okay. So they tried to do it a little bit three-dimensionally, and the very nice thing is what you have over here is where it's cut open from the side. You can also see those larger dots that you see here, the larger circles. That's the myosin, yeah? And the smaller dots, that is the actin. These are the thin filaments. I think this is pretty important here, also to know how it looks when it's cut open. Yeah? And here, down here, excuse me. You also have a different way of drawing this. Look, This here, what I'm coloring in here right now, that is supposed to be the myosin, the thick filament. Yeah. And yeah, that's the Z line. I don't know, titin, you don't have to know that, right? Yeah, and the actin, these are the thin filaments, and of course this here, that is the Z line. Yeah, and here as well, we've got the Z line, right? And these things here, that is the actin. Okay, and what I'll be doing now is yeah, let's have a look here on the left side. So this here is the actin. Yeah, so the here it is, actin. Okay, and this one over here, that is the myosin. And so you see, actually, when you look more deeply into the whole thing, you're going to discover that the actin, or rather the thin filament, maybe you should rather say the thin filament, is made of three things: so-called troponin, tropomyosin, and the actin itself. Yeah, so maybe that's something that we should write down here. The thin filament is actually made of three parts. The actin, which is the main part. We always refer to the whole thin filament as actin because that's the main thing here. Then you have troponin, which is another protein, and tropomyosin. Please don't confuse, there's the word myosin in here, okay? But tropomyosin is not the same thing as myosin, okay? So what you see here, that this is actually that is the myosin, okay? The thick filament, and do you see that there are little heads here? Little heads sticking out, yeah? These little heads that are sticking out are supposed to be looking in the other drawing here, yeah? These are little heads that are sticking out here on the side. I'll be talking about those later on again. And the question is now, of course, is why do you need to know these three things here? Yeah, because I'll be explaining this. Because the way that a nerve signal actually makes the whole thing contract, that is, yeah. You need an interplay between these three proteins, okay? So I hope that you're able to orient yourself a little bit better right now. The purpose of this series is simply to show you a little bit, yeah, different drawings as well, so that you can find your way around, okay? Yes, up. Why does it always jump back? Here we go. See, this one over here is a larger. I have a problem with this thing here. Okay, yeah, that is a thin filament here, okay? And this thin filament, so I'm going to change color again here. I'll change it back to white. So the thin filament which we always refer to as actin, actually is not only made of actin, but also of troponin and tropomyosin. You see over here on the left, thin actin filament. And the reason why I'm doing this here is because I would like to explain to you now what actually happens when a muscle contracts. Okay, so the control of muscle contraction is actually the main title here. Let me see if this works. Okay, so control of muscle contraction. Okay, when you move, where does this start? Well, it starts actually, of course, in the brain, because there is a nerve signal which travels down to the muscle. Okay, so nerve signal, we call it an action potential. This nerve signal, this action potential goes from the brain, of course, to the muscle. Okay, and what happens now is the following. When the nerve signal, so I don't know, how am I going to draw this? So AP stands for action potential. Yeah, AP, so that you actually know what this means. That's the nerve signal, and this action potential arrives at the muscle and causes a release of calcium ions, calcium Ca2+ or plus. Yeah, so that's these are calcium ions. Calcium ions are released by the nerve cell. Okay, and these calcium ions now diffuse, and they will now go to the troponin and they will activate it. Okay, so yeah, you see that the troponin is associated with the actin filament, yeah, and the calcium will activate this one. It will bind against it, and when it binds against it, then the troponin changes shape, and the troponin will then push the tropomyosin. Yeah, it's kind of, yeah, I don't know if the word push is the right word to use here. But it kind of will push the tropomyosin away a little bit. Or actually, in chemistry or in biology, we say it causes a so-called a conformational change, a change in shape. Yeah, so maybe push is not the right thing, but I mean, maybe you get the idea. Yeah, so the troponin kind of activates the tropomyosin, and what happens now is that this tropomyosin will then move. Okay, and it will then also shift. It will move, and by moving, it will expose on the actin here certain binding sites. So I don't know. I'm gonna draw. I don't know somewhere. I don't know if it's really there. Okay. So what I have here is yeah. I'm just gonna draw this as blue circles. These are so-called binding sites. For the myosin, which is the thick filament. Okay, so it shifts, yeah, and kind of pushes on the actin, yeah, shifts the actin, or if you don't like shifts, you can pushes the actin, maybe same thing. Okay, pushes the actin so that the binding sites for myosin, so that myosin can bind. Maybe, yeah. Maybe that is the better way to actually say this. Okay, so it shifts the actin so that so kind of pushes it away. Yeah, and basically what yeah, honestly, I put this in parentheses because I don't know if they're actually that frequent, the binding sites. So that's something that I'm simply guessing here. But that's not so much of an importance here. The idea is really that it causes the binding sites to be exposed. Okay, and then the myosin can push on the actin, and then the fibers are able to slide. Okay. So I don't know, this looks a little bit chaotic. So maybe what you can do is when you copy this down, that you have some kind of a a nice numbering system here. So that first we have the action potential arriving, then number 2 you have the calcium being released. Okay, number 3 the troponin is activated. The troponin activates the tropomyosin. The tropomyosin, so to say, activates the actin, and then number 6 is that when the actin is activated, the myosin is able to bind. Okay, so so basically myosin can bind. It's maybe number 6 over here. Okay, so you see that this is kind of a one thing leading to the next thing. Yeah. So that is kind of the idea of how muscle contraction is controlled. Okay, we're starting off with a nerve signal, yeah, and we're ending with a change in the actin so that the myosin is able to push the actin. Okay, yeah. Honestly, I have to admit I don't like what I've drawn here. Maybe I should take this away again. I don't quite like what I've drawn here, because I don't know if actually the binding sites look like this. Okay, yeah. It's also not so important maybe. Yeah? So yeah, what I'll do now is I'll move over again, and let's. Well, so this here is the microscope slide with the tongue of a rabbit, and of course this tongue contains a lot of muscles, so we'll be looking at this under the microscope, and this is a so-called a micrometer slide or a calibration slide, and it works a little bit like a ruler. So there are tiny lines on the slide, and the distance between two lines is 0.1 millimeter. So I'll also be taking a picture of this under the microscope so that we can measure out the size of a sarcomere. I just put both of these slides under the microscope, and then I took 2 pictures at the same magnification. Yeah, so what we have here is this is a picture that I've taken through the microscope slide. Through the microscope, no, it's a picture of a microscope slide, a so-called a micrometer slide. And you see from 4 to 5, how big is this? Let me change this again to white color here. So from 4 to 5 here, this here, as a matter of fact, that is 1 millimeter. And of course, then this basically means that those little units here from one small line, from one division to the next, that is of course 0.1 millimeter. In case you want to know how big this is if you have a dot of this size, okay, somewhere, then this is pretty much the smallest thing that you are able to see from a regular viewing distance of around 25 centimeters. So if you have a piece of paper in front of you with a dot that is 0.1 millimeter across, then you're just barely able to see it. So this is kind of the limit of what your eye is able to see. Okay, and what we would like to do now is I'd like to figure out what is the size of a sarcomere. Okay, so what I'm going to do now is look at this here, and this here is the muscle. Yeah, this red thing, that's the muscle of a small rabbit. The tongue of a rabbit is muscle, right? And just to help you a little bit orient yourself, you see that here the muscle fibers are kind of going like this, right? Yeah. And then you have some muscle fibers that are over here, that are on the left side here. Don't quite look like this, and the reason is because those fibers are kind of going out of your computer screen. Okay, so they're kind of extending outwards towards you, and the other fibers here, they're kind of stretching across like this. Okay, and those here, they are kind of moving out of the screen towards you, so to say. Okay, this is why they're kind of looking different, and also those over here. Yeah? So so that is basically where we would like to have a look now. And again, here is those things. Remember, these are the little, no, maybe not the best thing. This is not the way I should do this. Let's erase it again. So from here to over here, this is 0.1 millimeter. Okay, so this is 0.1 millimeter. So that's basically the thing that you're just able to see. But I would like to see now a so-called a sarcomere. So we still need to really magnify this much more. And look at this here. This here is now from here all the way to over here. I usually always use the edges here because it's easier to measure out. That is now this 0.1 millimeter. We're really magnifying it that much, okay? Because I always use it measure from the one edge to the other one, because it's kind of too difficult to find the center here. Okay, so that's why I never use the center, but actually an edge. Okay, so but that's basically what we'll be using here. And what you can do is you can actually do the same thing that I'm doing on screen. You will have different numbers when you measure it out on your computer screen or on your mobile phone, but you should get the same final result. Okay. So what we have now here, again, is a kind of superimpose this. So again, this is from here to here. That is our 0.1 millimeter from before. It's the same thing, right? And same scale, I really zoomed in now into the muscle of the tongue of the rabbit, and now you're able to see. Do you see this here? Light, dark, light, dark. You see those light and dark bands here? That's kind of cool, right? And I would like to measure out how big is this, because from light band to light band, so that's a sarcomere here, that's the second sarcomere, third sarcomere, fourth, and so on. Okay, but I want to know now how big is this in millimeters, right? So what I'm going to do now is I'm just going to take my ruler and I'm going to put it on screen right now. You're not able to see this, but I'm going to actually measure this out now. I put it directly on my screen. And the 0.1 millimeter is 158 millimeters on my screen. So it's different. In your case, of course, 158 millimeters. Okay, so if you are using a mobile phone, then it's probably going to be smaller. Or if you have a big monitor, then of course it's going to be bigger. So, but please also do that. Okay, try just write down 0.1. is yeah, 158 millimeters. So you actually might be, I'm just going to write this over here. Look at this. The 0.1 millimeter corresponds, yeah, to 158 millimeters on the screen. Some people write it like this. You can also write it differently. You can also say 0.1 millimeter, and you can write it in the form of a fraction, which is mathematically more correct. 158 millimeter. Just know here on the top that this one is the real scale, and this one is down here, the real size, and this one down here, that's the screen size on screen, the enlarged one. Enlarged. Okay, this is supposed to be enlarged, okay, and that's the real. Okay, so and now what you're going to do now is you're going to measure out. You don't change, you don't zoom in, you don't zoom out. It's really important. Using the same scale, you now are going to measure out in millimeters one sarcomere. Now, but be careful. Don't measure out one, but measure out 10 of them, and then divide it by 10, because this way it's going to be yeah, more precise. So what I'm going to be doing is you know what, I'm first going to erase this again here, simply for making it a little bit more visible. Otherwise it's too messy. So what I'm going to do now is I'm just going to start here maybe, and I'm going to count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So from here to here. You can also try to do this, and I have here, that is what, 29 millimeters on my screen. Okay. It is 29 millimeters. Again, you're going to get a different value because your size of the screen is different, and so on. Okay. So what we have now is the following: 29 millimeters. So this means that we have 2.9 millimeters for one sarcomere. Okay, on screen. So what I'm going to do now is the 2.9 millimeter that is on screen. So what I'm going to do now is I'm going to say X in real life corresponds to 2.9 millimeters. Again, if you don't like this way of writing, then I'm going to erase this here again. If you don't, then you can also write it down in the form of a fraction. So, and you have then over here X corresponds to 2.9 millimeters, and it does not matter whether you have the X on top or on the bottom, as long as you have on one side, this one over here should be the real one, and this one down here should be the enlarged one on screen. But if you flip them around, both of them, okay, then you get the same result. Okay, but you're not allowed to just have this one like this and the other one, the X, and this one flipped. That's not allowed. Okay, so I'm messy again here. Okay, so I just wanted to clarify this, and then in order to figure this out, what the answer is you cross multiply. So it is 2.9 millimeters multiplied by 0.1 millimeters equals 158 millimeters times X. So this times this equals this times this. Okay, and then you solve for X. I'm going to again erase this here. Okay. So what you can do now is the following. You simply divide each one here by 158 millimeters, and this cancels out. Of course, you also have to divide this by 158 millimeters. Look what happens. Millimeter cancels out here and here. We've got this millimeter over here left over. And when you type it into the calculator, what I will get for X is approximately 0.002 or 0.0018 millimeters, which is around 0.002 millimeters. Okay, how much is this? 0.002 millimeters. This is 2 micrometers. A micro sign is this U sign with this long line here. Okay, so this one over here from one sarcomere here is 2 micrometers across. Okay, 2 millionths of a meter. The question is now the following. Okay, how big is this now? You have to understand that the size of bacteria are approximately 1 to 2 micrometers. So if you've got a bacterium, yeah, it's about this size. Okay, a prokaryote. Yeah, so that is all sorts of different bacteria here. Okay, that is approximately the size. Okay, that you have here. By the way, there is also part of the syllabus. You have to know how to do those magnification calculations. Okay, so I'm not just doing this because I want to for fun, so to say, but you actually have to know how this works. Okay, so my advice is also you calculate it, and you should also get around 0.002 millimeters. I think when I typed it into the calculator, I got 0.0018 millimeters, if I remember correctly. Okay, so yeah, if you have something in that range, then you'll probably be fine. Okay, so let's move on. Here this is the empty foil where I normally would have done my calculation. These are the 2 more foils. And what I would like to do now is I want to talk about the mechanism of muscle contractions. I'm going to change the color again. Okay, the mechanism of muscle contraction. How does muscle contraction actually work on a chemical level? That is kind of the idea. And so first of all, let's make sense here of what we're seeing here. So the yellow one, that is the actin, or the thin filament. And those blue things here, that's of course the myosin, the thick filaments, and those things here that you see here, these are the so-called the heads of the myosin, the myosin heads. Myosin heads. And of course you can see that over here, those myosin heads, they cross-link and they bind against the actin. And when we go back a couple of foils, remember what I told you here, that myosin over here, so that myosin can bind. Yeah, that is the reason. Okay, the myosin heads should be able to bind. Okay, so let's go forward a little bit again. The question is now what happens. Okay, so I'm just going to erase this again here. Yeah, so that we have a little bit more space. It's one of the nice things about using a tablet like this here. And what I'm going to do now is I'm going to label the individual steps and actually what happens here. So first of all, what we have here is at the beginning is that the myosin, the myosin heads, they form cross links, or you can just say, why do we say cross links? They just bind to the actin. Okay. To actin. Okay, so that is you can see, that's basically this happens here. It's bound. The blue one is bound, the blue myosin is bound against the yellow actin over here. Muscle contractions, they need of course energy, and energy is in the form of ATP. And what we have here is this little star with the 3 phosphates. That is supposed to be that is here ATP. ATP stands for adenosine triphosphates. There's 3 phosphates connected here, and this ATP is energy rich. And you were able to see 2 things here. First of all, ATP has bound against the myosin, and look, there is no more cross-link. Okay, the head has kind of detached. So when so ATP binds against the myosin, and when it binds, as a consequence, the myosin heads detach from actin, from the actin. Okay, you can see that actually, right? So the myosin can either bind only the ATP or the actin. The myosin can bind either ATP or actin, but not both. Yeah? So, and when the ATP binds, then the myosin head goes away. So, number 3, just look at the drawing over here. What happens here? We're splitting off one phosphate. And when we're splitting off one phosphate, we are releasing energy. So, and what we have over here, the thing that's left over is known as adenosine diphosphate. And we have a PI. This is the so-called, the PI stands for an inorganic phosphate. It is simply the chemists and biologists simply are using PI as a an abbreviation for inorganic phosphate. And when you do that, you are releasing energy because between, I'm going back to the next step here, there is this bond between the second and the third phosphate, and that is a so-called a high energy bond. So this is where the energy is stored. Okay, so in this third bond, the energy is stored, and when we're splitting off the third phosphate, the energy is released. So ATP is converted to ADP plus PI. So it basically what means this means is energy is released. And every time when I say energy is released, you have to ask the next question is, where does the energy go? Okay, I mean, if I say it releases it, then we're using the energy to do something. The question is, what are we doing? Okay, look at the position of the myosin head. The myosin head, it's like a spring, okay, moves forward. Okay, so the myosin head moves forward, and this basically, this is where the energy is stored. Okay, and it's actually a little bit like a spring. You have to understand. Okay, it's almost like a spring, yeah, like a metal spring, and so it's kind of, you see, it's cocked into position. It's locked into position, cocked into position, storing the energy. Okay, so what happens next? Ooh, I already see that I've made a bad drawing. Over here, that is number 4. Look what happens to the head here, the myosin head. Myosin head binds against actin. On a further away position, okay, on a different position, further downwards. Further, should we say downwards? Okay, further, actually downwards might not be the correct one. Further towards the center, further towards further towards the Z line, actually. This would be the better way of actually saying it, okay? Because what we have is we want to move it to inwards, so the Z line actually it would be over here, right? Yeah. So we're going to remove this further towards the sides of the sarcomere, yeah? Sarcomere, okay? And look, last one, and this is where I've made a slightly bad drawing, so maybe you're going to copy it a little bit differently. You call this a so-called a power stroke. And this is when the energy is released, okay? So the ADP is released. Okay, it already contains very little energy anyway. And the potential energy of the head snaps backwards. Okay, ADP that's released, and the head, well, actually the power stroke is not the ADP being released, but actually the head snaps back to original position. Okay, the head snaps back to the original position. And the reason why you shouldn't copy it like this, you should actually maybe mention that the the actin moves like this. So actually, yeah, the actin, yeah, is now further down. Okay, to illustrate this a little bit, but I did not draw it like this. Yeah, so the head snaps back to the original position, and then we're back here at the beginning again. Okay, yeah, and we're able to do now the next cycle, and this is yeah, for me fascinating, because it actually sees how a large movement like muscle contraction is actually able to be seen also in the form of a chemical reaction. Okay, that happens. Look, guys. I think this was actually it. Okay, yeah. So what I've done in this short, more or less short lesson, I labeled a sarcomere. Okay, over here. I've given you a schematic diagram here of a relaxed and a contracted sarcomere. Then here we looked a little bit, I don't like it, it jumps around like this. We looked a little bit also on different ways of how it can be drawn, and also on how it actually relates to a whole muscle. Down here we have also different ways of actually drawing it, so you get used to slightly different ways of drawing it. Here, oh gosh. Yeah, here we also had a look at how actually the muscle contraction is controlled. So starting from a nerve signal, from an action potential, actually to the binding of the myosin. So and you see that you have a whole bunch of other proteins like troponin and tropomyosin also involved. So you see that one thing causes the other thing to happen. And then we did a size calculation over here. Where hopefully you should have gotten the same result as I have, even though you probably have a different sized screen. And finally we looked also at the mechanism of muscle contraction and how this happens on a chemical level. Yeah, I think that's pretty much everything that I wanted to say, and yeah, that's all. Wish you a nice day. Bye-bye.